Hello, hello, my dear students. I hope all have been doing well. Today, we are going to start our first lecture, and this lecture is about digestion and absorption of carbohydrate. This is the introduction, and after this lecture, in lecture number 2, we will be studying about the metabolism, catabolism, and anabolism of these molecules. So, why it is not going? So you all know that what are carbohydrates are sugars, and they are having formula CH. They are having carbon, hydrogen, and oxygen atoms. They can be found as monosaccharide, disaccharide, and polysaccharide. The major source of carbohydrates is plant, but carbohydrate that we found in our food, they contain they may be polysaccharide, disaccharide, or monosaccharide. Monosaccharide cannot be further metabolized because they are already simple sugar, and but they can be uh, and the disaccharide and the polysaccharide further hydrolyzed can be further hydrolyzed. Polysaccharide uh, means consist on polysaccharides are starch, glycogen, cellulose. They made up of more than 10 monosaccharide units. Disaccharide made up of two monosaccharide units, and uh, they are sucrose and maltose. So in this lecture we are going to see how this polysaccharide, disaccharide, and the simple sugar further hydrolyze. So what happens when these carbohydrates in the form of sugar come in contact with our body? So these carbohydrate metabolize the metabolism of carbohydrate start from digestion of carbohydrate, um, start from mouth, and from mouth, and it ends on intestine, and from mouth to intestine. Uh, Two type of digestion takes place. One is mechanical digestion, and second one is chemical digestion. So in this picture I will briefly describe you that in which part of the body what either chemical or mechanical digestion takes place, and what are enzymes that help in digestion of this carbohydrate. So in the mouth, when we take carbohydrate as a food in our mouth, what happened in this place, both chemical and mechanical digestion takes place. Our tooth chew the food, and this is called as mastication, and to break down food into smaller pieces, and this is the mechanical digestion. And however, the salivary juices that is released from the salivary gland mix with this small pieces of food, and further break down it, further digest it. So salivary actually in salivary juice there is an enzyme, that is amylase, that is present in this salivary juice. This amylase digests carbohydrates. So in the next slide we will see how the amylase digests food and where it works. But here excuse me, here we just have to understand need keep in our mind that in mouth there is a chemical and mechanical digestion takes place, and salivary juice help our mouth, tooth, and the salivary juice help in chemical and mechanical digestion. And after that this food reaches to the stomach. In stomach there is no any chemical digestion takes place, because in stomach means in salivary amylase cannot work in acidic pH of stomach. So here only mechanical digestion takes place by the peristaltic by the movement of stomach muscles. Only mechanical digestion takes place, and after that and the saliva gastric juices that release from the stomach mix with this digested food, partially digested food, and make a bolus that we called as chyme, and then this chyme will reaches to the intestine, and in intestine the further digestion takes place, and in intestine this chyme come into contact with the intestinal juice and the hepatic juice and pancreatic juice that releases from liver and the pancreas, and this hepatic juice, pancreatic juice and intestinal juice will mix with the chyme and then further digested it. Actually, this intestinal juice, hepatic juice and pancreatic juice containing different enzyme. We will study in the next slide what enzyme are present in this hepatic juice, pancreatic juice and intestinal juice, and on what part of the carbohydrate they act. We in this slide we have just keep remember that in intestine the juices, the enzyme that is present in the three type of juices come into contact with this partially digested food and further digest it, and then finally the absorption of the digested food takes place in our intestine. So this was the first outline of digestion and absorption, where and how the digestion and absorption takes place, and these are the overview here in mouth. Salivary this salivary juice that is released from the saliva, it contain enzyme alpha amylase. And then what alpha amylase does we will study in the next slide, but here we just have to keep in our mind in saliva an enzyme is present, that is alpha amylase, that help in the digestion, and stomach no enzyme release from the stomach, uh, and no chemical digestion takes place in the stomach, and both chemical and mechanical digestion takes place in mouth. And then in pancreas, pancreatic amylase released from pancreatic cells, and this pancreatic amylase further digest the food, and then intestine these different enzyme that together we call as dextrinase. These are five enzymes that release from the small intestine. These take part in further digestion in intestine. So what happen in mouth? In mouth, chemical and mechanical digestion takes place, and the uh, how chemical and mechanical digestion takes place in mouth. In mouth, the salivary gland release the saliva, and this saliva contain enzyme alpha amylase. We also call is this enzyme as petalin or talin, and this enzyme work on pH 6.7, and this enzyme need chloride ion for its activation. And what this enzyme does, here we can see in this picture, alpha amylase break down alpha 1,4 bond that is present between two monosaccharide unit that is found in a polysaccharide. For example, this food that we are taking, if it is glycogen molecule, it is a polysaccharide, and it is made up of a number of small monomeric unit that are monosaccharides, and these monosaccharides join via alpha 1,4 bond. So what this alpha amylase do, this alpha amylase break down the bond between two monosaccharide and then convert this polysaccharide into different unit of monosaccharide. For example, maltodextrin, that is oligosaccharide, then maltose, and isomaltose. So this is one of the big monosaccharide. This is one of the big carbohydrate that is oligosaccharide, that will contain 2 to 10 monosaccharide unit. But all these are small monosaccharide, um, uh, small sugars. Um, so what happened then? Food in from the mouth, this all happen in the our mouth by the action of alpha amylase. This glycogen molecule, that is polysaccharide, convert into oligosaccharide and monosaccharide. And then what happened in the stomach? In stomach, um, digestion stopped temporarily. And then what happened? Then only uh, stomach juices, that is gastric juices, mix with the this partially digested food and make it as a bolus that we call as chyme. um, why digestion did not takes place in the stomach? Because stomach, there is no carbohydrate splitting enzyme is available in gastric juice. So then food reaches in the intestine. And what happened in intestine? In intestine, uh, intestine release intestinal juice, that is containing different enzyme. And then intestine, pancreas, that release pancreatic juice. It is containing amylase, uh, again amylase, but this name is amylopsin. And this is different from salivary amylase. This amylase digests food. uh, how it digests the dextrin that reach from the mouth to the stomach to the intestine. It's further break down by the action of pancreatic amylase into small dextrin and the maltose and isomaltose. So this happen in the intestine. And uh, this digestion takes place in this intestine. And pancreatic amylase helps for this digestion. However, in intestine, the intestinal juice that reaches for the digestion of the carbohydrate, this intestinal juice contain different enzyme that collectively we call as dextrinase. And these enzyme are a family of enzymes that are containing 5 enzymes. And one is maltase, other is isomaltase, saccharase, lactase, dextrinase. Maltase, what do this maltase convert hydrolyzed maltose, that is disaccharide, into glucose, and 2 molecule of glucose. Isomaltase hydrolyze isomaltase, that is a disaccharide, in 1 molecule of glucose, 2 molecule of glucose. And saccharase hydrolyze saccharose, that is also a disaccharide, into glucose and fructose. And lactase hydrolyze lactose in glucose and galactose. And dextrinase hydrolyze limit dextrin. What is limit dextrin? Small dextrin into, this is not means small dextrin means it is contain few unit or 2 unit of dextrin, not it is not oligosaccharide that is previously containing 2 to 10 monosaccharide unit. So the dextrinase convert this dextrin into glucose, maltose, and maltotriose. So this happen in the small intestine. Small and uh, then when the this carbohydrate convert into maltose, saccharose, and lactose. And here we can see in this picture the maltase, saccharase, and lactase hydrolyze this maltose into glucose and sucrose, sucrase into fructose, so and then lactose into galactose. So only carbohydrate that reaches into the blood stream from the intestinal cell or intestinal wall are glucose, fructose and galactose, and these carbohydrate hydrate absorbed from the intestinal wall into blood. So how they absorb from the intestinal wall into a blood epithelial cell and then reaches into blood. Here two type of transport takes place, means these carbohydrate um, reaches in the blood by means of two type of transport. One is active transport and other is facilitated diffusion. So what is facilitated diffusion, what is active transport? In facilitated diffusion, the molecules of monosaccharides or molecules of sugar transport via channel protein, via carrier protein, from higher concentration to lower concentration, means towards the along with the concentration gradient. We say this along with the concentration gradient when molecule transport from higher concentration to lower concentration. However, in active transport, what happened, this sugar molecule move from lower to higher concentration by the help of some transporter protein, and this is against the concentration gradient that requires energy in the form of ATP. So what happened in is intestinal, how these glucose molecule transport via active transport from the intestinal wall to the epithelial cell, and then into the blood. They actually here in the intestinal wall, this is a picture of the luminal epithelial brush border cell. So here we different transporter protein found. What they do, they transport glucose against the concentration gradient, but they against the concentration gradient and along with the other minerals like sodium. So this is the we say this type of transport, in that sugar or other molecule transport with different other minerals, means if two molecule transport together, so we say this co-transport or uh, co-transport or symport. So here there are seven type of glucose transporter that form different part of our body, that GLUT1, GLUT2, GLUT3, 4, 5 and S SGLT1. So these molecules are glucose transporter that form different places in our body, or and they help in the absorption of glucose. And the question is this: why our body is not be able to digest the cellulose? Cellulose actually are polysaccharide, and they are made up of beta 1,4 glycosidic bond, and human body is not containing the enzyme that can break beta 1,4 glycosidic bond. So this is the reason that why cellulose cannot be hydrolyzed or cannot be digested in our body. When we take cellulose, it did not digest, but it is remain in our intestine as a form of fiber, and then it excrete out from the body. There are different diseases that occur due to the inability of human body to digest different type of sugars. For example, if human body not able to digest mm, lactose so to glucose, so a condition takes place that we called is lactose intolerance, and this disease is occurred due to the deficiency or absence of lactose enzyme. Lactose is an enzyme that is responsible for the breakdown of lactose into glucose and galactose. So what happened, this lactose accumulate in your gut, and then what happened in gut bacteria, in gut the bacteria do fermentation and convert this lactose into H2 and carbon dioxide. And what happened due to this H2 and carbon dioxide, means concentration increases in the gut, and there will be acidic environment, and that due to this high concentration of H2 and O2 and acidic environment, water absorbed from the intestine to the into the gut, and it do the dehydration and the diarrhea. So this is the disease that occur due to the um, absence of the lactose. And here is another disease, that is sucrose deficiency. This is the same disease, means this disease is occurred due to the deficiency of also lactose enzyme, but it occurs in the um, human. This deficiency is occurred due to the this sucrose deficiency is occurred due to the absence of sucrase enzyme, and it is occur in children. And sucrose enzyme actually able to hydrolyze sucrose into glucose and fructose, and due to the deficiency of sucrase, the sucrose cannot hydrolyze or digest. And but this deficiency is occurred due to the absence of sucrase enzyme, but it is having same symptoms as lactose intolerance. So these are the some references. Thank you very much for your kind attention.